Lipid Peroxide Fluorometric Assay Kit. Fluorescence Microscope Experimental Guide. Elabscience Lipid Peroxide Fluorometric Assay Kit. 1. Suitable for LPO assay of aderent cell. 2. This video shows the process using fluorescence microscope for detection. Unpack the kit, check the label and items. Read the full instruction before use. Please contact the supplier with any question. Reagent preparation. Preparation of buffer working solution. Mix well 45 ml of double distilled water and 5 ml of buffer. Mix fully. Five micromoles per liter probe solution. Mix well 4,995 microliter of buffer working solution and 5 microliter of 5 millimoles per liter probe. Mix fully. Protected from light, and it should be used up within 2 hours. 100 micromoles per liter positive control solution. Mix well 4,995 microliter of buffer working solution and 5 microliter of 100 millimoles per liter positive control. Mix fully. Protected from light, and it should be used up within 2 hours. Sample preparation. Plate setting. Operation process. Remove the cell culture medium and rinse the cells gently using buffer working solution. Add buffer working solution to blank well. Add probe working solution to control, positive control and experiment well. Incubate the cells at 37 degrees with shading light for 60 minutes. Removed working solution. Add 1 milliliter of buffer working solution to wash the cells 2 to 3 times to remove the probes that did not enter the cells. Measurement. Add 10 microliter, 0.01 mol PBS buffer to the slide. Remove the cell sliver and place it on a slide. Fluorescence microscope parameters were set. FITC or GFP channel was selected, and the slide was mounted on the microscope for detection.